Good morning, I'm Doug Dollimore. Welcome to this news briefing from the 252nd National Meeting and Exposition of the American Chemical Society in Philadelphia. We're joined today by Dr. Mary McCourt, Dr. Lawrence Mielnicki, Jamie Catalano from the from Niagara University. I got it down. We're so used to saying University of something, but so. Anyway, they will be talking to us about their work on an oral insulin pill for the treatment of diabetes without a shot. Dr. McCourt?
Okay, the way this all started was a few years ago. We were looking at trying to understand how to deliver drugs to the brain. So we started looking at the materials that were being used, and we said these don't seem to be working well. And rather than making an incremental change, we decided to try new materials. So we used these neutral lipids, natural materials, and what we found is that we were able to create these little vesicles, these nanoscale vesicles that we've called cholestosomes. And when we started to study them, we realized that we could make them any size. The surface was neutral. They survived a really wide range of pH. And we said, it sort of dawned on us at that point that this could be something that could be used to deliver things orally. So, of course, one of the things we decided to tackle, we've tried to tackle many different kinds of things, but we have looked at insulin as a potential orally available drug. And we have, in fact, found that we are capable of encapsulating insulin in these little vesicles. These little vesicles get into cells. The rodent studies indicate to us right now that they are orally available. They are systemically available, whether IV or oral, and they survive intact. The insulin survives intact through the GI tract and then passes through and is delivered intact and gets into the bloodstream.
Dr. Mielnicki, do you have anything to add?
Yeah, well, the interesting part of, in addition to insulin, we can package anything inside these vesicles and potentially deliver a lot of therapeutics. And so there's a lot of applications for this technology beyond insulin. And so we're very interested and happy to see the success in the initial animal studies that we've done with insulin.
Okay. And Jamie?
Hi, yes, and I know that Dr. McCourt and Dr. Mielnicki really summed up the materials and biological aspects of what we do, and my role really is to incorporate this technology in the lab. And I'm just lucky because I have professors that, you know, come in and work with us every day, so I was able to learn a lot.
Okay. Are there any questions? Okay, Kath?
So it's Kath O'Driscoll from Chemistry and Industry magazine. I guess we've heard a lot over the years about an insulin pill, and I know there's one already in clinical trials, as you mentioned. What makes you think that this one will actually succeed, and why is there a need? If there's already one in clinical trials, what's different about this one?
Do you want to tackle that from a biological perspective?
Referring to the Oramed medication, yeah, they're actually starting phase 3 trials with that. The important distinction is that we're packaging in a nanoscale vesicle, whereas they're using a different type of composition, sort of like an emulsion. And they're passing it through in an enteric pill. So they're very different mechanisms of delivery. And I think in their initial animal studies, they saw 10% availability at best, and our initial animal studies seem to be indicating we're a little better than that. So there's always room for improvement, and I think we have some improvement if we can adapt this technology for delivery.
Can you say how much better? I mean, that was another question. What is the bioavailability you've seen?
We've been probably conservatively, we can say we're from that figure 2 to 3-fold better at least.
Okay. Also, just to ask, what is the lipid that you're using? What is this naturally occurring lipid? And how is it able to survive these conditions in the body?
Well, they're cholesterol esters. And so they're naturally present in the body. The body uses them for lots of things, so they're not recognized by the body as something different, and then I think in the industry term, they're generally regarded as safe.
But the acid conditions in the stomach, though, that's...
Once the vesicles are packaged, since they're neutral, they don't interact with any charged environment, and so the acid in the stomach has no effect on them.
Okay, thank you.
Ben, you had a question?
Thank you. Ben Valsley, Chemistry World. One thing I was wondering, thank you for clearing up what the cholesterol esters themselves were, but is there any interaction between the insulin itself and the lipid, or does it essentially just remain encapsulated in the middle?
We've got some evidence that, if in a general sense, encapsulating things is an interplay between what we're trying to encapsulate and the lipids. So to some extent, there is an interaction, and that somewhat will affect the final composition of the product.
But it doesn't denature the...
It doesn't do
insulin or
anything
anything like
that affects the actual construction of the vesicle, so to speak. We will get them.
Excellent. Well, I think Kath took all the rest of my questions. Thank you very much.
Okay, we have an online question from Christine Suh, Office of Public Affairs. How long does it take for your insulin pill to have an effect, and how does that compare with a shot?
Go right ahead.
Well, we probably should clarify that
It's not a pill.
this is not a pill. So what we're giving, in the animal studies, we're giving it orally in a liquid volume. And so the action that we've seen is similar to that of the controls. That's what I can say. I mean, it really, time-wise, it's very similar. And there's some evidence that we have a longer acting effect.
Okay, and I have a question. Katie Cottingham, Office of Public Affairs. So why do you think it's taking so long to get toward a non-injectable type of insulin, be it a pill or a liquid that you would drink or something like that?
I mean, it's for the two main reasons that it's been a problem since insulin was first discovered. The fact that it gets degraded rather easily, and we can't get proteins in general across the intestinal membrane. It's just a very hard problem to solve. We can solve getting it through the stomach because you can always put it in a pill that will travel through the stomach and dissolve in the intestines, but then that second problem rears its head, and it's a hard one to get over.
Kath?
I'm just wondering about the process to encapsulate them in the liposome. How easy is it to do that? And also, what would this kind of cost? How expensive would it be?
Jamie's our excellent student formulator, so she would like to speak to the actual physical process of doing that.
Okay, well, one thing to also clarify, I know we talk a lot about how the cholestosome is actually not a liposome. There's...
Very important.
Yes, it's a neutral lipid-based vesicle. It's not charged. And can you repeat the second part of your question?
It was really about the process to actually encapsulate it to make the liposomes.
Yep.
They're not liposomes.
Cholestestomes, yes.
Cholestestomes,
But the process includes solubilizing the cholesterol esters first, and then once we remove the solvent, they're left as a nice film that we then sonicate in the presence of our insulin solution, where those insulin molecules are then encapsulated, or a cocoon of lipids is formed around the insulin.
Okay. How about dosing? I mean, what sort of doses are in these pills as well?
The animal studies were using small doses, typical doses that are given.
A unit per kg.
1 unit per kilogram is
Is
for
in
the
our
animal.
studies.
Yeah.
And again, just to ask about the sort of expected cost, do you think is?
That's a hard one to answer because we have the materials themselves and the time, and we haven't really gone to scale up because we're still at the lab scale and discovery sized preparation stage, and so we've got enough to do small scale studies in animals, but at any one time we don't have a lot. So putting a cost on it is hard to say, but the materials themselves are widely available and generally inexpensive compared to some other things that might be out there.
There's some mention, I think, at the back in the kind of abstract about solubility, that this might have been a problem with formulating these.
One of the things that because of the way that their formulation is done, we want to have as high a concentration of the molecule in the aqueous component. So what Jamie's work was doing was to optimize the parameters that will allow for the maximum solubility of the insulin in the aqueous component, which allows for the maximum encapsulation in the vesicle itself then.
Right, thank you.
Ben?
Thank you. As the vesicles are uncharged, do you foresee there being any problems in terms of keeping them in suspension for long-term storage and delivery when it does become a medicine?
We've got various and sundry observations on that issue, but in general, they stay suspended, and when suspension becomes an issue, there are things that you can add to help keep them in suspension. So we haven't really had a problem when that issue presents itself. It works out the same way as others have pretty much done it with the same excipients and things.
We find the vesicles to not be they're not leaky. So once they're inside and protected, they're not likely to come out. They don't just passively come out.
But the lipids themselves don't coagulate?
No,
Okay.
that's correct.
Based on your animal studies, do you foresee that this new approach might be universally applicable to patients who need to take insulin, or is this just for a subset that it only works within a range?
If it turns out to be effective in animal studies, then yeah, it would be effective for any situation where insulin dependence was a problem.
Katie Cottingham again, Office of Public Affairs. So what other conditions might you treat with these cholestosomes? Or is this just a diabetes treatment?
No. We've encapsulated very small molecules to very large molecules like IgG. One of our other students is presenting a poster on an IgG kind of molecule. And so we're looking to the animal studies indicate that we can get into the brain with these things, so we're looking for any sort of brain delivery. We have some evidence that genetic material can be encapsulated as well, and to be able to turn on those kinds of things, it would be very helpful to us, I would say.
There's a wide range, and I'd like to think that almost every condition that has a treatment for which there's a drug, we can go ahead and encapsulate that drug.
Yes, we can.
Are there any other questions? Well, thank you. The archived version of this session will be posted soon at bit.ly/ACS Live Philadelphia. Please join us for our next press conference today at 10:30 a.m. on using houseplants to clean indoor air. Thank you.